Dr. Raphael Stricker has a degree in internal medicine from Columbia University in New York with subspecialties in hematology, oncology, and supplemental training in immunology and immunotherapy. He is an internationally recognized leader in the diagnosis and treatment of tick-borne diseases. He is a long-time board member of LymeDisease.org. He's past president and board member of the International Lyme and Associated Diseases Society, and he serves on the Lyme Disease Advisory Committee that advises the California Department of Public Health. He's in private practice in San Francisco. He has authored over 200 medical journals and abstracts and serves as co-principal investigator of the MyLymeData project. Dr. Stricker?
Thank you, Dorothy. Let's see if I know how to work this thing. It's a pleasure to be here. It's great to see such a great turnout. Let me move this up a little. Last time we did this, which was about 7 years ago, I believe, we had a very good turnout too. So it's good and bad that we have this much interest in Lyme disease. It shows that we probably haven't made as much progress as we should have, which isn't surprising. So first I want to say that I have no financial conflicts to disclose. Particularly, I have no links to any laboratories that do testing for Lyme disease or treatments for Lyme disease. So the talk that I'm going to give you is a talk that I originally put together for a large pharmaceutical company. And I thought that it would be interesting to get big pharma interested in Lyme disease. And I was trying to figure out how we could do that. And I think that this talk is important for patients to understand too, because the same factors go into pharma interest in Lyme and patient interest in Lyme. What I'd like to do is show you the problems that we have with Lyme disease and why Lyme disease is really very controversial. Lyme disease has been called the new great imitator. After seeing this slide, maybe it should be called the new great imitator. But it mimics syphilis, which used to be the great imitator because it could mimic any disease in medicine. And I'll show you some of the problems with this mimicry. Now, Lyme disease, as many of you know, is caused by a spirochete, which is a corkscrew-shaped bacteria called Borrelia burgdorferi. And here you see a picture of a corkscrew-shaped spirochete in culture. But this is actually not the Lyme spirochete. This is, in fact, Treponema pallidum, which is the spirochete that causes syphilis. And it's very hard to tell the two apart. As you can see on the slide, the syphilis spirochete has a few more turns than the Lyme spirochete, but otherwise they look pretty much the same. Lyme disease is transmitted primarily by Ixodes ticks, but this is not an Ixodes tick. For those of you who know anything about ticks, you can see that it has this white dot on the back. So this is actually an amblyomma tick, which is also called a Lone Star tick because of that white dot. Now, amblyomma ticks have expanded tremendously in their distribution around the US. So when you see those maps from the CDC that have, a little bit in the Northeast and the upper Midwest, that's just ridiculous because look at this tick. It has a huge range. It's basically taken over the whole country. And amblyomma ticks can transmit things like Ehrlichia and Rocky Mountain spotted fever. They do carry the Lyme bacteria, but they've never been proven to transmit Lyme disease. So that is kind of a problem. If you're a purist, you can say, well, these aren't a problem because they don't transmit Lyme disease, but they transmit a lot of other things that are a problem. Now, one other very recent fact that's come to light, this is a fossilized tick from 20 million years ago that was found in the Dominican Republic, and this article was published recently. This tick was found in amber, and it is in fact an amblyomma tick, just like the one that's been spreading all over the US. And when they looked inside this amblyomma Lone Star tick, they found blood cells from a monkey, and the monkey blood cells had evidence of Babesia microti infection, which is a co-infection that's transmitted by ticks. So here's a tick from 20 million years ago that was feeding on mammals and could transmit a tick-borne disease. So these ticks have been around a long time, and they're not really going away. Now, many of you know that the hallmark rash of Lyme disease is the erythema migrans or bullseye Lyme rash. However, this is not an EM rash. This is a rash that was in a patient who said, this is the most itchy rash I've ever had. I've never had anything as itchy as this. What could this be? This is actually a case of ringworm. And a lot of times, unfortunately, Lyme rashes will be misdiagnosed as ringworm. So what you have to do is you have to ask the patient, does it itch? And if the patient says, yes, it itches, then it probably is ringworm. If the patient says, no, it doesn't itch, my wife noticed it when we went to bed last night, then it's probably a Lyme rash, because they usually don't itch. Now, Lyme rashes also are not always bullseyes. They can have different shapes, different sizes. They can be solid. They can look like a bruise, like the one here on the lower right. So again, a lot of confusion over what's a Lyme rash, and this creates controversy over Lyme disease. Lyme arthritis, which is the classic symptom of Lyme disease, which was described in Lyme, Connecticut in the 1970s, causes big swollen joints. But this is actually not Lyme arthritis. This is arthritis due to Staph aureus, which can look just like a Lyme arthritis. Lyme arthritis is actually quite uncommon. Only about 20% of Lyme patients will ever have true arthritis with a swollen joint. What Lyme patients do get, as many of you know, is they get musculoskeletal pain. And this can be migratory pain. It can affect almost any joint, any muscle group. It can move around. It can be very weird. And when you go to the doctor and tell them this, they think that you're kind of crazy. But this is very typical of Lyme disease. Now, neurologic complications of Lyme disease, such as Bell's palsy, which is facial nerve palsy, which you can see in this patient, is actually kind of uncommon in this country. It's much more common to have neurologic complications of Lyme disease in Europe, although we do see some patients who develop Bell's palsy. But the typical Lyme patient in this country is much more likely to look like this. Yes, George W. Bush had Lyme disease, which is a fact that he covered up pretty well while he was president, and he really hasn't talked much about his Lyme disease. I think someday he will, and it'll be nice to hear exactly how he was treated for his Lyme disease, which he got on his ranch in Crawford, Texas. Other famous people have been diagnosed with Lyme disease, author Amy Tan, Hall of Fame pitcher Tom Seaver, folk singer Pete Seeger, and rock and roll star Daryl Hall have all been diagnosed and treated for Lyme disease. Other famous people, Richard Gere, Avril Lavigne, the guy I can never remember on the right who plays Donald Trump on Saturday Night Live. I think of him just as Donald Trump now. They've all had Lyme disease and been treated for it. And then Allie Hilfiger, who's the daughter of clothing designer Tommy Hilfiger, had Lyme disease, and she was diagnosed in a mental hospital and actually treated for the disease and got all better. And she wrote a book about her experience called Bite Me, which you should all read because it's really pretty good. She was treated with intravenous antibiotics and got better, fortunately, from her neurologic disease. A couple of other famous people, Yolanda Foster, from a famous housewife, has been very vocal about her fight against Lyme disease. A picture that might surprise you is on the lower right, Michael J. Fox. As you know, Michael J. Fox was diagnosed with Parkinson's disease at the ripe old age of 30. And he has raised millions of dollars for the Parkinson's Disease Foundation. What most people don't know is that before Michael J. Fox developed Parkinson's disease, he was diagnosed with Lyme disease. He got Lyme disease at a film shoot in Canada. And he claims he was treated for the Lyme and got all better, and it has nothing to do with his Parkinson's disease. But that's kind of an interesting coincidence. So that brings me to the topic I'm going to talk about, which is the new pharma frontier for Lyme disease. You know, what can pharmaceutical industry do for this disease? And in order to understand we have to define the scope of the problem, develop better testing, and develop better treatment for Lyme disease. And the goal is to implement precision medical care for Lyme disease patients. So first, defining the scope of the problem. Lyme disease, more than 300,000 cases of Lyme disease caused by the spirochete Borrelia burgdorferi are diagnosed each year in the US. Thus, Lyme disease is 6 times more common than HIV/AIDS, 20 times more common than hepatitis C virus infection, and 30 times more common than tuberculosis in the US. Ixodes ticks carry more than 237 bacterial strains, and new strains of the Lyme spirochete, such as Borrelia miyamotoi and Borrelia mayoni, are being discovered almost every day. Birds play an integral role in the wide dispersal of Borrelia burgdorferi infected ticks. Rodents, including mice, shrews, squirrels, and chipmunks, are the primary reservoir of Borrelia burgdorferi. I think I saw a squirrel outside that was carrying around a Borrelia. Culture of viable Borrelia spirochetes in human genital secretions suggests that Lyme disease could be transmitted by intimate contact from person to person, and I'll get back to that in a minute. So that brings us to this statement from one of the most prominent medical journals in the US, the New York Times, and it was written by two members of the Infectious Diseases Society of America who said there is no credible scientific evidence for the persistence of symptomatic Borrelia burgdorferi infection after antibiotic treatment. So when we look for credible scientific evidence in the medical literature, what we found is a lot of studies from Europe and the US that show persistence of the Lyme bacteria after antibiotic therapy. And these are studies that have been done actually this is only up till 2011, so there have been 6 more years of studies like this. And it's obvious that there is such a thing as persistent infection and infection that fails antibiotics, the current antibiotics that we have. So that's really why we need big data, like what Lorraine was talking about. And just to show you some first studies of this, this is a study from Johns Hopkins University where they took a database of 47 million people who were enrolled in a range of US commercial health insurance plans. They identified over 547,000 of these patients, and then they focused on over 52,000 of these patients. So this is really big data. And what they found was that over 63% of the treated Lyme disease cases had at least one diagnosis associated with, quote, post-treatment Lyme disease syndrome, which is really chronic Lyme disease by a different name. So basically, about 2/3 of Lyme patients fail conventional antibiotic therapy. So it's not 5%, not 10%, it's 2/3 of patients fail this therapy. And this is really a disaster for current treatment for Lyme. And what happens to these patients? Lorraine showed you this slide in a different form, but they get really sick. So how many of these patients have fair or poor health? On the right, you see 16% of the general population that estimates that they have fair or poor health. On the left, 72% of these patients have terrible health after getting Lyme disease. And this is more than all the other diseases here: congestive heart failure, stroke, heart attack, diabetes, multiple sclerosis, arthritis, liver failure. These patients are sicker than almost every other category of patient with chronic disease. So this is a big problem. And as one patient said in the film Under Our Skin, which, by the way, you should all watch if you haven't seen it already, Lyme disease usually doesn't kill you, it just makes you wish you were dead. And that's the state of the art today. Now, going back to the infectious disease experts, this is a statement from the CDC website that says there's no credible scientific evidence that Lyme disease can be spread from person to person through sexual contact. But in fact, nobody's really looked at this very much, and there are some studies in animal models showing sexual transmission in mice and in dogs in very elegant studies. And actually, interestingly, in ticks, ticks can transmit the Lyme bacteria by sexual contact. Interestingly, there are some inbred animal models like Lewis rats and hamsters where there is no sexual transmission, so it does seem to depend on what the animal is like and the breeding of the animal. So now what about humans? This is a study that was done by Marianne Middelveen, looking at couples having unprotected sex who had Lyme disease. What the study showed was that there were live Lyme spirochetes found in genital secretions, and when the spirochetes were characterized, they had the same strain of the spirochete in their genital secretions. Interestingly, there was one couple, though, that had a different strain, and only they had that strain. None of the other couples did. So this again suggests that there is transmission from person to person in these couples. And in the pictures worth 1,000 words category, this is a semen sample from one of these patients. It shows 2 Lyme spirochetes on the left and a sperm cell on the right. So again, there is this contact or a suggestion that there's an association with sexual transmission. So Lorraine showed you all those questions that are important to Lyme patients. It would seem that a top priority question would be, can Lyme disease be transmitted by intimate human contact? Interestingly, when Zika virus was discovered, it took the CDC about 10 seconds to conclude that Zika virus was sexually transmitted, but when it comes to Lyme disease, we really don't care. So, second category, developing better testing. Testing for Lyme disease, as many of you know, is very controversial. The two-tier test system that the CDC endorses and the FDA involves a screening enzyme-linked ELISA or immunofluorescence test followed by a confirmatory Western blot. And this is the test system that's been in place for about 30 years. So the problem with this test system is that the CDC and FDA-approved commercial two-tier testing has a sensitivity of only 46%, which means it misses more than half of Lyme cases. And in contrast to that, if you look at HIV testing, it has a sensitivity of 99.68%, so it misses less than 1 in 200 cases of HIV. So Lyme testing from a sensitivity point of view is inadequate. Now, you'll hear a lot that the specificity of this testing is great. It's 99%, so there are very few false positive tests. But a study recently showed that in non-endemic areas, the specificity of this testing is as low as 20%. So what that means is that this testing, commercial Lyme testing, is essentially worthless for most Lyme patients. And the high cost of commercial test development and haphazard regulation have hampered improvement in Lyme testing. And better testing is available, but it needs to be accepted by the medical establishment. So what does this testing look like? This is a comparison of the CDC testing with IGeneX, the IGeneX system for testing Lyme. And IGeneX did a very simple alteration of the CDC system. They used a different target. They used 2 strains of Borrelia instead of the 1 strain in the CDC system. And they used different interpretation criteria from the CDC. And what you can see here, if you look on the upper left, the CDC system has a sensitivity of 77%, and if you only look at IgG, which is what they say you're supposed to do, the sensitivity is 49%. So again, it misses half, a little more than half of Lyme cases. Now, in contrast, if you look at the IGeneX system, which is in the lower right, you get a sensitivity of 97%. So 49%, 97%, which one would you rather have? It's kind of hard. The specificity of IGeneX testing, if you use a confirmatory test, is also around 97%. So this is really a very good system for diagnosing Lyme disease, but unfortunately it's been rejected by the CDC and many other specialists, and this is a problem. If people would actually accept this, we would have a pretty good diagnostic system for Lyme. There's a novel culture test for Borrelia that you've probably all heard about. This was developed by Ava Shoppy's group in New Haven. The culture test has a sensitivity of 43% in a week. So within a week, you could diagnose about 43% of Lyme cases. So it's about as good as two-tier testing, but it's also much more expensive. And it's unclear whether this system will survive the criticism from the CDC and others. The company that makes this test is trying to get FDA approval, but right now the jury's out on this. Now, what other approaches to diagnosing Lyme are there? Well, there's been a lot of interest recently in what's been called companion diagnostics. So companion diagnostics are not direct tests for the infectious agents. They're tests that are associated with the infection. Let me show you a couple of these, which we use routinely. One is looking for the CD57 natural killer cell subset, which is a subset of white blood cells that is suppressed in most patients who have chronic Lyme disease. This has been a very good marker for the disease, and it's useful for monitoring patient response to treatment. And again, it's not really accepted by most of the medical establishment, but that's their problem. The other test is C4A testing, which is an inflammatory marker that's associated with chronic Lyme disease. Again, not a specific test for Lyme, but it's very helpful in seeing how patients respond to treatment and how the treatment is working. Now, other groups, especially academic groups, have been more interested in cytokine expression, which is shown on this slide, and I expect you all to memorize this slide because it's really, really important. So cytokine expression, as you can see, is very complicated. And actually, what's interesting about this slide is that when you see a report that says, we found the one thing that's the most important thing in Lyme disease, this is the most important marker for Lyme, think of this slide. This is how complicated the immune system is in response to Lyme, and there is no one marker that really tells you what's going on with Lyme disease. So this has been a problem in terms of using cytokines as markers for the disease, but you'll probably hear more about this from academia. All right, then finally we come to better treatment for Lyme. Current treatment for Lyme disease features antibiotics that are 50 to 70 years old, such as penicillins, tetracyclines, cephalosporins. As I told you before, about 2/3 of Lyme disease cases treated with 2 to 3 weeks of these antibiotics develop symptoms of post-treatment Lyme disease, so they basically fail these ancient treatments. Studies of prolonged treatment, quote unquote, for chronic Lyme disease used 90 days as a maximum, which is inadequate compared to other chronic infections that require anywhere from 6 months to 5 years of treatment or more to eradicate the infection. And different forms of the Lyme bacteria, pleomorphic forms, biofilm colonies, and antibiotic-tolerant persisters of the Lyme spirochete make treatment challenging. And just to go through this very briefly, pleomorphic forms are different forms of the Lyme spirochete. They can be spiral on the upper left, blebs, cysts on the lower left, or biofilm colonies. And this is work from Leona Gilbert's group in Finland, showing that the forms have different effects on the immune system as well. So it becomes a very complicated system with these different forms of infection. Biofilm colonies have been looked at by Eva Schappe's group. Different strains of Borrelia have different forms of biofilms, which are basically aggregates of the Lyme bacteria covered with slime, and the slime protects them from antibiotics and the immune system. It's not really clear how important biofilms are in Lyme disease, and we can talk about that later if you're interested. But certainly the other forms like cysts and resistant forms seem to be very important. And then antibiotic-tolerant Borrelia persisters have been studied by Kim Lewis in Boston and Ying Zhang at Johns Hopkins. And these are not strictly resistant forms of the bacteria. These are forms that just tolerate antibiotics. And if you don't give enough antibiotics, they just sort of sit there until the antibiotics go away, and then they start growing again. So this is actually a big problem for this disease. So how can we get around these different forms of the Lyme bacteria? So looking at other chronic diseases that have been addressed by big pharma, you come to the conclusion that there are certain targets in these organisms that make very good targets for treatment of the disease, and particularly HIV disease and hepatitis C virus infection has had these specific proteins called proteases that are very important in the life cycle of these viruses, and these have been the targets of novel treatments that have resulted in significant advances in treating these diseases. Now, Lyme disease has the same kind of target, so why can't we target these things in Lyme disease? So what I'm going to do is I'm going to talk about 3 of these potential targets. There are a lot more, but these are 3 that have been looked at to some extent. Ying Zhang's group did this study. This is actually the best study he's done, looking at what happens to these tolerant Borrelia organisms. And in fact, what he found in these organisms, one of these proteins is upregulated 30-fold, and this is called the CLIP protease. The CLIP protease has this structure. It's actually a barrel-shaped protease. It has an entry point, an exit point, and then the protease in the middle. So what does this CLIP protease do? Well, it's kind of a meat grinder or a garbage system for Borrelia. It actually takes misfolded proteins, unprocessed proteins, protein aggregates, chops them up and gets rid of them. So one approach to treating Lyme disease would be to block this protease, and that has never really been done. Kim Lewis had the bright idea to do the opposite, to kind of open up this system and just make it, function indefinitely. That actually did not work in Lyme disease, so that approach was not successful, but blocking this protease could be very successful. So that's one target. Another target is the HTRA protease, which is shown here. HTRA protease is very interesting because it's a multifunctional protein that is involved in protein processing and also invasion of Borrelia into other tissues. And in fact, this has been so interesting that it's even been patented. And the patent belongs to, I don't think I can read it on this slide, but it says, the government of the United States of America, as represented by the Secretary of the Department of Health and Human Services, Centers for Disease Control and Prevention. So yes, the CDC has a patent on this protease. That's how important it is. What they intend to do with it, I really don't know. The third protease that I want to just highlight is in the RNA helicase family. And this is a very interesting family of proteases because it includes the protease that is the hepatitis C virus protease that has been the target of all these spectacularly successful drugs for hepatitis C. Now, the Lyme bacteria has a similar protease that's shown here. And this protease actually is interesting because the gene for this protease appears to qualify as what's been called a boss gene. Maybe they should call it a Trump gene now. So this gene can actually regulate a lot of other genes, and in fact, it regulates 187 other genes in Borrelia. So it's a very, very significant gene, and also it can regulate infection of mice and tick transmission of the Lyme bacteria. And so this would be a very obvious target for treatment for Lyme. So there are a lot of these proteases in the protease menagerie, which is shown here. This is actually from plants, because plants have a lot of these proteases, and we'll see how we can do that at some point. So in the last 2 minutes, I'd like to talk about the carrot and the stick. Again, I told you this talk was for pharma companies, so what's the carrot, what's the stick in this? So the carrot in developing novel treatments for Lyme disease is the market for these drugs. So if you look at HIV/AIDS, about 55,000 new cases a year. The market for treatment of HIV, $18.5 billion a year. Hepatitis C virus, only about 13,000 new cases a year in the US. The market for that, $4.5 billion. So Lyme disease, with over 300,000 new cases a year, what would the market be for drugs that treat this disease effectively? So that's something to think about. So that's the carrot. What's the stick? Well, patients with Lyme disease are figuring out that the CDC isn't going to help them, the NIH isn't going to help them, the FDA isn't going to help them, the Infectious Disease Society of America certainly isn't going to help them. So who is going to help them? It's the pharmaceutical industry. And this is exactly what happened with HIV/AIDS. You started having demonstrations and trashing pharma booths and die-ins in the pharma parking lot. That's the bottom slide. So eventually, I think Lyme patients are going to get to this level of activism, and then pharma companies will have to do something to address the disease. And I've got 1 minute left, so I'll show you my conclusion slide. The scope of the Lyme disease epidemic is much larger than HIV/AIDS, hepatitis C, and tuberculosis, and may involve sexual transmission. We need better testing using companion diagnostics for Lyme disease. We need better treatment for Lyme disease using designer drugs similar to the ones that have changed the course of HIV, AIDS, and hepatitis C. And we need big pharma to take the lead in the global fight against Lyme disease. And then finally, my field of dreams. You can see the ticks are out there, but build better drugs, and they will come. Thank you very much.